Every second you are awake, toxic metabolic waste builds up inside your brain. It is a biological inevitability of consciousness that requires a nightly, specialized clearance operation. Neurons require a highly regulated environment to communicate effectively, relying on precisely controlled levels of potassium and calcium. But the continuous act of firing signals generates toxic byproducts. If these chemical leftovers, which include proteins like amyloid and tau, linger in the tissue, they become neurotoxic. Left uncleared, the brain's cognitive infrastructure degrades. To remove this debris, the body relies on the glymphatic system. This is a microscopic plumbing network of perivascular tunnels, tightly wrapped by the end feet of specialized glial cells called astrocytes. Surviving the high metabolic cost of consciousness requires active, mechanical maintenance. The brain has to literally wash itself to stay intact. Cerebrospinal fluid, or CSF, constantly bathes the exterior of the central nervous system. It pools around the brain's entry points, waiting for the right conditions to permeate the tissue. The entry gateways are aquaporin 4 water channels. These microscopic pores sit directly on those astrocyte end feet, dictating exactly when and how fluid moves into the brain. These water channels do not operate continuously. Their function is strictly governed by the brain's internal circadian clock. During the mid-rest phase of sleep, AQP4 expression highly polarizes. The channels physically align on the vascular structures, allowing cerebrospinal fluid to efficiently cross the boundary and enter the tissue. Falling asleep acts as a precisely timed mechanical trigger. The clock signals the astrocytes, the channels align, and the floodgates open. While researchers have mapped the hardware of this self-cleaning network, exactly how the fluid navigates the dense brain tissue is currently fiercely debated among biophysicists. One camp argues for bulk flow, where arterial pulsing forcefully drives fluid and waste straight through the brain tissue. But others point out the extracellular space's high hydraulic resistance, suggesting the system relies instead on diffusion, where molecules slowly migrate across tight gaps. Whether driven by advective pressure or steady diffusion, the biological imperative remains the same. The metabolic waste has to exit the tissue to prevent neurological decline. Theoretical physics aside, the observational data is clear. The clearance rate of toxic proteins is demonstrably higher when a subject is asleep compared to when they are awake. This happens because the physical architecture of the brain changes. During slow-wave sleep, the interstitial voids between brain cells expand by 60. Basic fluid dynamics dictates the result. This geometric shift enhances the waste clearance rate by over 2.5 times. Sleep physically morphs the brain's internal architecture, widening the gaps so the accumulated trash can escape. To see what happens when this process breaks down, researchers recently turned to non-invasive MRI scanning in community-dwelling older adults. This diagram illustrates researchers tracking microscopic flow using the DTI ALPS index, measuring water diffusivity along perivascular spaces. The scatter plots reveal a clear negative correlation. As sleep quality drops, marked by severe sleep apnea, glymphatic functioning significantly stalls out. Events like sleep apnea cause temporary hypoxia, or oxygen desaturation. This disrupts the microscopic fluid flow, trapping the toxic proteins inside the tissue rather than flushing them out. Chronic sleep disruption acts as a physical blockage in the brain's infrastructure, leaving older adults with measurable accumulations of neurotoxic waste. When the cleaning infrastructure fails, the physical connectivity of the brain itself begins to stiffen, directly impacting how the mind retains information. This diagram shows structural and functional network coupling. In healthy brains, functional communication flows flexibly across physical pathways. But without clearance, communication hubs become rigidly locked together. As mapped below, for poor sleepers, this locked network state directly correlates with steep memory decline. While human imaging provides correlational evidence, the biology is clear. Depriving the brain of the time it needs to clear debris guarantees neurological damage. Your memory's maintenance schedule relies on this nightly rhythm. You have to go to sleep so your brain can go to work.